So yesterday we saw a presentation about air eDNA, and there's a lot of presentations about aqueous eDNA, but where are the soil and the sediment people? So for this presentation, I'll be focusing to another subgroup of eDNA, which is sedimentary DNA, and hopefully I'll be able to convince you how this one is particularly useful in reconstructing past and current biodiversity assemblies in different aquatic ecosystem. So I work with a lot of indigenous partners, so I'd like to start my presentation by doing this acknowledgement, territory acknowledgement. So the entire study was conducted within the Treaty 8 territory, the traditional lands of the Cree, the Dene, and the unceded territory of the Métis people. I'm currently affiliated with University of Victoria, so I'd like to acknowledge the Lekwungen peoples, whose traditional territory Yuvik stands, and the Songhees and the Esquimalt and the Husanich peoples, whose historical relationship with the land continue to this day. And before I start with my actual presentation, I'd like to acknowledge this amazing group of people who helped a lot in making the different aspects of this project possible. It's a highly collaborative project, so there are eDNA people, there are sediment chemistry people, and we just combine everything and then see whatever story what we can generate out of this data. So this particular project was funded by the Oil Sands Monitoring Program. I was supported by iTrackDNA project and at the same time by the Libero Postdoctoral Fellowship. And these are like the different institutions involved in this project. So as we all know, we are experiencing rapid biodiversity loss, and I'll be focusing on freshwater fishes. In terms of number, 36% of the global freshwater fishes are now classified as threatened according to the IUCN Red List. And I think part of the reason why we are all here right now is that we acknowledge that eDNA can be an alternative approach to augment conventional survey methods so that we can have rapid and accurate diversity monitoring data collection. And I'll be focusing on lake sediments. What's fascinating about lake sediment is that it serves as a natural biological archive that basically consists of bacteria, a lot of plant pollen, fish DNA and fish artifacts. You also have a lot of zooplankton and a lot of phytoplankton communities preserved properly in between the layers of the lake sediments. And each layer will correspond to a specific time zone, so that will enable you to reconstruct past and current biodiversity changes. Since I'll be focusing on lake sediments, I'll be talking a lot about sedimentary DNA, or I'll be referring to as sedDNA in this presentation. I'd like to emphasize that this one is not a novel approach. This has been used by several researchers all over the globe, but basically they are focusing on phytoplankton communities, zooplankton communities, or even bacterial and plant communities. In terms of fish, or in terms of metazoan in general, I think we're still having substantial problem when it comes to detecting fish or mammals when it comes to sedimentary DNA. Based on metagenomic data, so basically these are all sequences of sedimentary DNA, metazoan communities will only constitute around 0.3% of the total sedimentary DNA. The rest, 99.7, it's basically zooplankton, phytoplankton, plant communities, and bacterial communities. That's why it's really challenging to detect fish when it comes to sedimentary DNA, and hopefully I'll be able to address that one in this presentation. So to give you an idea, basically most of the sedimentary DNA comes from aqueous DNA. It's basically released by the organism to the water column, and through sedimentation process, they settle at the bottom. And once they are in the sediment, basically your positively charged sediment particle binds with your negatively charged DNA, thus allowing long-term preservation of DNA within your sediments. And that's basically the main mechanism how these things are working. So that even they are 200 years old, or even the oldest record I think would be the study in Greenland, which is around 2,000 years ago. We were still able to detect this DNA. And for this presentation, I work a lot with sediment core. Once you're dealing with sediment core in combination with sedimentary DNA, basically you can reconstruct the past 200 years. If you want older, the past 2,000 years ago or 5,000 years ago, it's just a matter of how deep your sediment core will be. But for this presentation, I'll be focused on the pre-colonial period. So that's basically around 50 centimeter topmost layer of the lake sediments, because I'm dealing with, or I'm in partnership with a lot of indigenous communities, and they're basically interested in reconstructing the pre-colonial period. So the questions that I'd like to address in this presentation: how can we improve fish sedimentary DNA detection? Because to be honest, it's really hard. Imagine you're detecting that 0.3% in your sedimentary DNA. How can we combine this particular approach with another knowledge stream of biodiversity data, in particular the indigenous ecological knowledge? And the last one that I'd like to address would be, can we reconstruct fish settlement histories with sedimentary DNA? In particular, I'm interested if this particular method will be capable of determining which one is native, which one is introduced in a certain aquatic ecosystem. So the first part, basically, I collaborated with the Sedimentary Asian DNA Society. We built a global network of people who are working with fish sedimentary DNA. And the first output of that network would be a review, hopefully to answer questions related to fish DNA detection in sediments. So, in collaboration with 26 different experts all over the globe, we basically consolidated 29 studies. And what we want to know is that what are the basic challenges that we encountered in processing this kind of sample. There are some success stories, but there's still a large portion which are unsuccessful in terms of detecting fish DNA in sediments. And these unsuccessful stories, these are the primary reasons that we can attribute to that one. One reason would be sediment sampling location. For paleolimnology, we basically go to the deepest portion of the lake because that serves as the reservoir. Or can you go to the limnetic zone? So where do you need to collect the sediments? We also attributed it like to detection method, whether you are using next generation sequencing or qPCR. Primary issues, amount of sediment inputs, like it ranges like from 0.5 grams to 20 grams. PCR inhibitors, there's a lot of PCR inhibitors in sediments because of the organic content. And then DNA extraction kits. And to answer these questions of like, why is it so hard to detect fish DNA, we basically need to go back to the ecological aspect of fish sedimentary DNA. What are the factors affecting the detection and at the same time the dynamics of this DNA in the sediment? One of the primary factors affecting would be the terrestrial input, because basically these are organic content that serves as PCR inhibitor. Another factor that we'd like to highlight would be the oxygen content in the sediment. It's somehow correlated with the bacterial degradation. You also need to consider the time of residency for the sedimentary DNA, because the longer it stays in your sediments, the more fragmented it will be. And when you're designing primers for this one, you need to target shorter amplicons as compared to 300 base pairs or 400 base pair amplicons, because basically you'll get highly fragmented DNA because of their age. Another factor would be bacterial degradation itself. More thing, I'm working with lake ecosystem, so this is not a big concern, but if you're working with like marine ecosystem wherein there's current disturbing your DNA dynamics, you need to consider the flow rate. How will the DNA be affected by the flow rate as it settles from the water column to the sediments? This one is important for me because fish biomass highly affect the shedding of the DNA. So basically, the higher your fish biomass and the more stable the population is, the higher the probability that you'll be able to detect fish sedimentary DNA. And the other factors are like the physicochemical parameters. For example, UV exposure, the pH, and the temperature within the ecosystem. And considering this ecological aspect, one part of our review is that we need to enumerate some methodological consideration on how we can improve fish sedimentary DNA detection. So these are the questions that we want to highlight. So for example, I need to emphasize the location for where you collect your sediment. Will you go to the deepest portion of the lake, or will you go to the shallower portion of the lake? Or can you collect on both sides? Another thing is that how many replicates do you need For sediment core, and then you're processing 50 centimeter deep, basically that's around 50 samples for one sediment core. So should you extend it into triplicates? Another thing is that if you're doing replications, do you need to pool the sample and how it will affect the entire data? Another consideration would be related to DNA extraction. For example, the QIAGEN Power Soil Kit would encourage the use of like 0.25 grams of samples for extracting DNA from the sediments, but there are some kits that are very specific in accommodating larger volume. For example, the maximum I've encountered would be 20 grams. And theoretically, the more sediments you input, the higher the probability that you can get fish sedimentary DNA. You also need to consider several steps of removing PCR inhibitors because I must tell there's a lot. There's tannin, there's phenolic compounds, and all possible PCR inhibitors will be present in your sediments. You also need to do an integrity check. For Helbing Lab, our standard protocols that we're using integrity DNA assay for checking the integrity of our sediment or of our DNA samples from the sediments or even from aqueous matrices. Another question would be, what kind of detection method should be used? qPCR is highly sensitive and it's very useful if you're targeting one or species of particular fish species. But the current trend in the field is that as much as possible, they want to use metagenomics to avoid PCR biases. It's also possible to use metabarcoding. These are like NGS-based approaches. So it depends on the question that you want to answer. Another thing is that how can you avoid false positive detection? Because you need to consider you're only detecting that 0.3% in your sedimentary DNA. So how can you delineate background noise from actual positive detection in your sample? So you need to have particular method on how to avoid false positive result. And then what are the bioinformatic pipeline if you decided to use metagenomics? Because you will be overwhelmed by the data that you'll be getting. So as part of the review, we also tried to recommend some possible things for troubleshooting. So first thing is that you need to consider fish biology. If you're detecting a limnetic fish, then collect your sample in a limnetic zone. If you're collecting a benthic fish, then go collect in the deepest portion of the lake. So you need to consider fish biology, and that will give you answer when it comes to where to collect your sediments. Another thing is that large volume extracted sediments would be highly encouraged. As much as possible, use the 20 grams of samples. But if you're working with sediment core like me, 20 grams is quite impossible to get. So right now I'm using like 2 grams to 5 grams of sediments for analyzing my samples. Another thing is that if you're using 20 grams of DNA, it will basically tell you to elute your DNA in 2 milliliter of elution buffer, and that's a lot. So what we are recommending is that once you elute it in 2 ml of elution buffer, try to reconcentrate your DNA so that your template will also be concentrated. Another thing, remove all possible PCR inhibitors using different methods that you know. It can be magnetic beads approach, or it can be like column-based approach. For primer design, we're very specific into targeting shorter amplicon region. As much as possible, we want to limit it lower than 150 base pairs because you need to consider the fragmentation of your DNA. And then increase or double the template of your DNA when you are setting up your PCR reaction. And this is the last part of this first part. We can explore augmenting conventional paleolimnological proxies for estimating abundance. So for paleo studies, we're using a lot of proxies such as fish individual counts, historical capture records, otoliths count. They are preserved in the sediment, so you can count them manually, and then even plankton abundance. So to complement these conventional methods for paleolimnological studies, sedimentary DNA would be really useful. And then we're also encouraging normalizing the DNA count using a degradation pattern of the chlorophyll in your sediments, where we found out that once you normalize it in consideration with the degradation pattern through time, you tend to observe better correlation between abundance data, quote-unquote abundance data, and your sedimentary DNA count. So I'm not telling you it's good for absolute abundance estimation, but it will give you an overview of the historical changes in the abundance. Yeah, so I think for this part, I'd like to say that knowledge of fish sedimentary DNA and its dynamic in the ecosystem, it's still on its infancy. We're still expecting a lot of improvement when it comes to this approach and when it comes to its proper integration in environmental monitoring. It's not perfect, but there are a lot of potentials. So I'll go with the next part, wherein I'd like to augment indigenous ecological knowledge with sedimentary and DNA inferred data. But I'd like to clarify that we're not comparing which one is better. We just want to showcase how the immense potential of once we combine these two methods, like what are the data that we can get and how comprehensive it will be. So it should be complementary and it should not be compared like which one is better. So as an overview, you have the oil sands region in Alberta, and I think we're all familiar that this one, just like other industrial activities, it's expected to affect total biodiversity within the area. Within oil sands region, we observe like a special scenario, especially Copper Lake, and in collaboration with the Chippewayan Prairie First Nations. This lake is quite special because the community, they have the detailed record of fish assembly in the past and also like current fish assembly. Based on their account, the lake used to have abundant lake whitefish species, but all of a sudden it disappeared after 1950s. So this kind of account can be hard to validate, especially when we try to check the conventional survey method from the Alberta fisheries. The earliest record that they have would be around 1960s. So before 1960s, we cannot validate any account. And this is where sedimentary DNA can be very useful. So for this one, we went to Copper Lake, go to the deepest portion, and then we collect sediment core. Each layer of sediment core was like divided per 1 centimeter interval, and then we analyze it using lead 210 for the dating of each sediment layer. I'd like to emphasize the importance of the assay validation process when it comes to this one. I'll always go back to the 0.3 value of fish DNA in your sedimentary DNA. And this is where careful and robust qPCR assay can be very useful. In Helping Lab, we're following the Lang-White workflow wherein we always start with the mitochondrial genome, explore the entire sequence, and then use unique-seq to identify a unique region to make your primers and probes very specific to your target organism. We also do genomic DNA validation in the lab so that we can test the specificity of the primer and the probes, and at the same time, sensitivity parameter characterization using G-blocks, and we're using EloQuant for this one so that we can establish the limit of detection and the limit of quantification. And to tell you, I'll show you how important the limit of detection and limit of quantification of the latter results. So for the assays, we use integrity DNA for viability or for integrity checking. We also use general fish assay that can detect general fish region for screening whether there's presence or absence of fish DNA. And we design assay for lake whitefish and Cisco. These two are highlighted because these are like the species of interest for the Chippewayan Prairie. Remember the account of lake whitefish before 1950. And then we use northern pike as positive field control, and we have walleye as positive or negative field control for this one because walleye is not present in Copper Lake. So to guide you with the structure of data that I'll be showing, first is the result of integrity DNA assay for the chloroplast detection. We detected it all throughout the sediment layers. Not surprising that we're getting the highest concentration in the younger sediment layer because they are more preserved properly and they're not yet fragmented. And then for this one, we use 4 technical replicates per reaction, and we're getting 4 out of 4. So this gives us more confidence that like the integrity of the DNA that we got is like we can use it for the downstream analysis. Another result would be, so you have limit of detection and limit of quantification. All of the values that we got are above limit of detection and limit of quantification. Another result would be the general fish assay. For this one, I'd like to emphasize that we use 16 technical replicates for the reactions because we want to increase the probability of detecting the fish DNA. Where we tried to use 4 and 8, but basically we're detecting like 1 out of 8. So we tried to use more technical replicates and we decided to use 16 for this. And yeah, we're detecting fish DNA all throughout the sediment core, from the oldest to the most recent layer. And for the specific fish that we want to target, we were able to detect the positive field control, which is northern pike. We were able to detect one of the whitefish species, which is cisco. There's no detection for the negative control, which is the walleye, and there's no detection for lake whitefish. For this one, this is where the limit of detection and limit of quantification would be very useful. We're getting background noise when it comes to the detection using qPCR, and our approach for this one is that if the DNA copies is above limit of detection and above limit of quantification, we're confident that it's more of a positive result or a positive detection. If it's lower than the limit of detection, then we'll just declare it as a background noise. And then, as you can notice, cisco, it was detected 1928 to 1948, so we feel the urge like to explain. It's present and then all of a sudden it disappears. So we want to reconstruct the past environmental condition. And we tried to do that one using sediment chemistry. So as you can notice, there is an influx of organic matter on the shaded portion, which is the presence and disappearance of lake whitefish. So there is a rapid increase in organic matter in the lake, and at the same time, there's also an increase in inorganic content. In correlation with the organic content, we also measured sulfur. That will give you an idea on the microbial respiration process. So there's also an increase in microbial respiration process during this time period. And as you can imagine, if your microbial respiration is increasing, they are degrading all this organic content and they are basically depleting your oxygen concentration. And that may be the possible reason why your lake whitefish suddenly disappeared. Another thing, in correlation with your inorganic content, we tried to measure calcium, strontium, and barium. That will give you an idea on the photosynthetic rate during this time period. And we basically saw increased in strontium. And this will give you an idea that there is an organic content increase. There's also possibility that there's a lot of algae in your water. So it's possible that there's also algal bloom during this time period. And based on this data, maybe that's why lake whitefish suddenly disappeared. We also tried to explore another knowledge stream, which is the indigenous knowledge. I'm not the one who collected the data. We just consolidated everything. We have an expert dealing with like indigenous communities. And they collected the data from the elders of the Chippewa and Perry First Nations. And before I explain this entire figure, because basically this will summarize everything for this chapter, I'd like to emphasize that we process our sediment samples in a randomized form. We implement a randomized processing number in our lab, so we don't know which layer is the first layer, which layer is the next one, and we'll just be surprised once we consolidated everything. So this figure shows you the year for the lead 210. It ranges from 1905 to 2019. It also gives us an idea of the sedimentation rate for the organic content contribution. So there is a rapid increase in organic content during 1940s. I'm also showing you where we detected general fish DNA using the eFish1. I'm showing you the positive control, which is northern pike. It's distributed in all layers, from the oldest to the most recent one. What surprises us the most would be the detection of cisco, one of the lake whitefish species. highly correlated with that story that the indigenous communities are telling us. Like, it used to be present in the lake, and all of a sudden it disappeared. So it somehow correlated during this time period, like 1950s time period. We also tried to relate it with historical land use. During 1910 to 1960s, there is rampant exploration for oil within this region, so there's a lot of human migration. There's also an account from the indigenous communities that there used to be a natural landslide on the eastern part of the lake that might also contribute to the increase in terrestrial input. You also have the most recent industrial activities within the region. It's related to forestry, logging, and especially the oil sands activities. Yeah, so basically, based on that figure, I can tell you that the combined use of sedimentary DNA and indigenous ecological knowledge can be a very effective approach in providing you a more comprehensive data when it comes to fish communities or whatever organism you want to test. And this is the last part. Reconstructing fish communities in different lakes. So basically, we collected DNA from other lakes in the region. I'll be presenting data for the 4 other lakes. This is the summary of the different fish species that we collected, and then showing you the viability testing or integrity testing, general fish, and then positive control and negative control for each lake. I'll be going back to your Copper Lake data. Seeing the unseen. We detected cisco that used to be present in this lake, extending the native range of particular fish in each lake, because if you're seeing a particular fish species from 1905 to 2019, that is more likely a native species because it's been there for almost 100 years. And that will give you an idea which one is native, which one is introduced, and when is the time period wherein they are introduced. And yeah, supporting conventional surveys, because checking the Alberta fisheries records, the earliest record that they have for northern pike, for example, it's been 1960s. But sedimentary DNA can give you the past 500 years ago. The same data that we observed in Burke Lake and Marie Lake, in comparison to conventional surveys, sedimentary DNA data predates the records. We also detected fish DNA from Winifred Lake, but this is a special case because the conventional survey, they have a record of like 1960s. And then we don't have record before 1960s. But we were able to detect different fish species in this lake. And then, yeah, for Cold Lake, this is interesting because it can give you native species idea, and at the same time for this lake, you have records of human-mediated introduction. They are seeding lake trout and rainbow trout in this lake for the longest time. They started 1,500 individuals, but all of a sudden, during 1960s, I guess, they decided to dump 300,000 individuals. And that's also the time point wherein we first detected signals from lake trout. So it's possible that using sedimentary DNA, it can give you an idea of human-mediated introduction within lake ecosystem. And using this approach, it can provide higher temporal resolution when it comes to building or reconstructing fish assembly within aquatic ecosystem. And to summarize, it's not a perfect method. It's still being developed, but there's a lot of promises. And I'll end my presentation by putting it in a bigger picture. Like, what's the future of fish monitoring programs? Well, it depends on the question that you want to answer. So you have conventional surveys, you have aqueous eDNA, you have sedimentary DNA, you have ecological indigenous knowledge. It's just a matter of pinpointing what's the strength and weakness of each approach. But ideally, we want to promote integrative approaches wherein combining all different knowledge streams when it comes to building biodiversity data in aquatic ecosystem. And that's the end of my presentation.